I'd like to thank everyone today to our webinar, which we will be discussing best practices and technology for fine kinematic movement and gait analysis. Just like to give a quick overview of the program we have for you today. Our first presentation will be given by Dr. Dilip Verma, who is the Moderator Product Manager for TSE Systems. He will be discussing the Moderator and telling you how it's the game changer in movement analysis and gait evaluation for kinematic insights. Our second presenter is Dr. Carmelo Bellardita, and he will be discussing behavioral profiling for dissecting brain function in health and disease. After each session, we will have a Q&A session. So if you have a question during either presentation, please type your question in the chat feature, and we will address those questions after each presentation. After both presentations, I will just have a quick wrap up of the program with follow-up links to be included. So TSE Systems worldwide, we are a global company offering products and services for preclinical research. We have our global headquarters located in Berlin, Germany, with home offices also in Chesterfield, Missouri, which is just outside St. Louis, and also in Beijing, China. We also have a global network of distributors and partners worldwide that help service our customers as needed. TSE Systems has over 133 years of experience offering products and services in the preclinical marketplace. Our facility in Berlin has over 4,000 square meters of production space to build it and design products. We have systems and applications in over 2,000 institutions worldwide, and we have over 10,000 different TSE systems in place. So our field of scientific expertise, TSE Systems offers products and services that helps researchers in 4 major research areas. First of all, we have our IntelliCage, our multi-conditioning systems, and the Moderator to help in behavior assessments. In the area of physiology, we have our stellar telemetry, which can record a wide variety of physiological parameters in animals ranging from mice all the way up to moose. In metabolism, we have our PhenoMaster system, which is an automated home cage system for phenotyping research. And we also offer a complete solution for both in vivo and in vitro inhalation exposure studies as well. So with our product overview in the area of telemetry, again, we can record a wide variety of pressures, activity, temperature, and biopotentials. For behavior studies, our products can do things that all sorts of conditioning studies, activity and exploration, anxiety, depression, motor function and performance. And in the area of metabolism, with our PhenoMaster, we can do things such as indirect calorimetry, food and drink intake and analysis, activity monitoring, and much more. For the types of home cage testing we have available, we have our single housing capabilities with our PhenoMaster system. We also offer group housing capabilities with IntelliCage. This is where you can house up to 16 mice together and do cognitive and behavioral assessments in a social setting. And then we can even go ahead and combine the PhenoMaster and IntelliCage together in a single system, which we call PhenoWorld, which allows you to have a total colony housing concept for your research applications. So I will introduce our first presenter today. I'd like to introduce Dr. Dilip Verma. Again, he is our Moderator Product Manager for TSE Systems. Dilip obtained his PhD in neuroscience at the Medical University of Innsbruck, where his research interest was looking at the pharmacological role of different neuropeptide wide receptors in fear and anxiety disorders. So Dilip, I will give the floor to you and let you tell everyone about the Moderator system.
Thank you very much. Thank you, Gary. Can you see my slides?
Yes.
Thank you, Gary, for the nice introduction, and welcome to all for our webinar. And today I will guide you why moderator is a game changer in terms of kinematic gait analysis. Moderator is a state-of-art instrument that utilizes partial automatization to evaluate fine motor abilities in a standardized, quantitative, and objective way by incorporating kinematics elements that target particular body regions. For example, iliac crest or anatomical parts like a iliac crest, hip, knee, ankle, or toe, where you can basically measure the distance between the two anatomical points or the angle inward or outward between the 3-point or the 4-point anatomical regions. And being said that all these anatomical coordinates you can directly correlate with the gait cycle, with the stride, both phases, the stance and the swing on each frame. And you can correlate whatever other coordinates being like a distance or angle, acceleration, velocity, all together, you can correlate with the whole stride cycle. Why we need to do kinematic gait analysis? As you can see that the gait disturbances may be caused by very wide range of conditions, including neurological disorders, Parkinson's disease, ALS, Huntington or multiple sclerosis, for example, or any musculoskeletal injuries, or the spinal cord injury, or simple ataxia or pain model, which is severely deviate the motion cycle. Or you can simply study the side effects of any drug, or recovery or the treatment efficacies, or simple aging effect on moderator or any rare neurological disease. And why we have to do this kinematic gait analysis, as you can see that we, in the end, want to provide a better therapeutics in the clinics, so the patients can have a normal life, which is they are, at least with these neurological disorders, are limited. So to do such kind of studies, definitely you need some devices to do preclinical studies. And as you can see, in the earlier time, we were having only Rotarod, which was having a limited readouts, for example, latency to fall. But this is not enough to do the fine kinematics analysis. Therefore, you need such a device like moderator, which can rate all these motorcycles in all the small laboratory animals, for example, rat mice or salamander. Moderators bring you a lot of unique features, which is no other devices provide in the market. For example, you can have a video recording from the 3 different sites simultaneously. So you can have a ventral recording, and you can also get left and the right profile through the mirrors in one video. Moderator also comes with a sophisticated software which called TSE Motions, and it's analyzed both, so gaits through the paw printing and also the fine movement kinematics, adding with this all anatomical parts, anatomical regions, hind versus forelimb in the left and the right profile. You can get on top these pattern analysis in ventral and the side view. Also, you can do this both in the dry and the water surface, and with the 4 different modalities. So you have a runway, which is similar to like a ground walk. You can have a horizontal ruts, so ladder walk, which is used for the fine kinematic analysis, or the wading module. For the wading, basically, you are using the same runway module. For the mice, you can fill the water up to 1.5 centimeter, and the 4 rats for roughly 3 centimeter. And then the animal is showing this wading movement, means walking torso high, means you can measure the muscle strength. You can see the muscle strength, how they are walking, right? Or For those animals who cannot carry the body weight over the ground, you can use the glass basin. So you fill the water in this glass basin and put your animal, severely motor disabled animal, into the water, and water supports the body weight. And you can measure the fine stroke movement. You can track them. So this is a really very unique feature of the moderator, which is in the market. And why moderator? Because we provide you with both paw printing as well as including the body anatomical parts, means the whole full body profiling, what you can do in moderator in the different mouse models, either the neurological disease model or any injury model. So for example, you can see this nice study with from Preissig et al., where they have used 3 different kind of mutant mouse models for ALS, Huntington disease, or cerebellar ataxia, where you can see direct comparison with these trajectories, how they are visually different from each other. Having all these anatomical markers on the left and the right profile, some of the markers shows the early detection of the symptoms before even the disease progress. For example, max hind toe height, or the front and the hind limb coordination changes you can detect at P30, so postnatal day 30. So which is as early as you can test an animal on the any behavioral testing equipment. So which is not possible in any other classical assay like Rotarod or the grip sprint or any other conventional motor testing. So this is very a unique feature on the moderator, so you can detect very young animals. So all in all, moderator gives you an opportunity to have a fine kinematic gait analysis on not only on the paw printing, but all the body parts. With that, you can have an early detection. You can have a videos on 3 sides, and based on these anatomical markers, you can create more than 100 parameters. And this is highly sensitive because as you can start with these few parameters as early as P30, right? And also you can combine moderator with the TTL box with measuring different biopotentials, for example, electrophysiology, so EEG recordings, or optogenetics and telemetric recording. Our second speaker, Carmelo, will also show you in his presentation some of these kind of measurements later. Moderator do have a very specific unique principle, working principle. So basically, and it's very simple. So you can shave the animal body, put the animal anatomical marks, and the software can track all these anatomical marks on each frame. And you can have roughly 300 frames per second based on the camera you are using. So we are providing 2 different cameras, 5 megapixel or 2 megapixels, so which can achieve by both. We can achieve highest frame, depends on what is your requirements. We can have this discussion while we are the sales procedure. And all together, basically you mark the anatomical parts and you are ready to track through the software. So still now, as I said, this is marker-based process, but we are also working on artificial intelligence, so markerless tracking. So being said, that means you don't have to mark animal body at all, but this artificial AI can detect all these anatomical parts automatically. And this is on the beta phase 2 at the moment. And some of the users maybe are also attending this webinar who is already using this AI, and this is will be available soon once we can reach the highest amount of accuracy. But with both the marker tracking or with the markerless tracking through the AI, you can directly track all these anatomical points either on the mobile animal, or you can also create later on a stick view, which you can also put an animation. You will see in my later slides that how you can have a visual clear differentiation between the 2 different genotypes, just putting this animation on this stick view. Based on these all anatomical markers, you can create several parameters. So general spatiotemporal parameters, so just the stride length or the stance and swing phase or inter-limb coordinations, diagonal cadence or left or the right alteration rhythm, swing phase and the path trajectories. So you can create with this stick view the trajectories of the movement, which you can directly analyze or can have a visual impression. Body posture, so being on these anatomical parts, as you can see here, the iliac crest height or the shoulder height, you can also, or the tail tip can be also a marker to create parameters, or the gait variability or deviations, for example, retraction and protractions, which is calculated with the static anatomical point and the full retraction and protraction of the hind limb or the forelimb. So such that you can create more than 100 parameters to have it for your study design. This is very nice publication by Prasik et al. In 2016. They have published, they have studied, I have showed you before as well, the 3 different mouse lines, so ALS or Huntington and the cerebral ataxia. And here they show this superoxide dismutase modifications in this gene, where they have studied temporally progressive changes in the hind limb movement, especially in the ankle anatomical region. So they can measure the ankle angle or the range of motion, ankle height, and the hind toe position with reference of iliac crest. And you can see that they have differentiated or significantly found the differences in the wild type and the transgenic animal in the earlier or the late stage of their age. Some of the parameters can be also detected much earlier, as I said before, like a postnatal day 30 or 40, for example, the tail height or the maximum hind toe height or the paw contact between the front and the hind paw. And so these are the symptoms which you can see much earlier than the full disease progress. So motor they give you an opportunity to have the to detect or to test the animal to have these early symptoms, so you can start the treatment. So this is an advantage of using moderator. So not only the so the anatomical parts, but also you can detect or track paw prints. So this is very nice study using moderator done by an accordion group with Yang et al. They have quantified here the diagonal paw contact interval. As you can see how diagonally the front right or the hind left or the front left with the hind right, they position, and they find a significant difference with wild type or the tau mutant mouse line. And this is also a very nice study published in 2010 in Nature Methods by Jonah et al., where they have showed the full 4 modalities of moderator by using rats. I won't go much in detail in the data, but I just want to show you how you can see the clear differences. So they have then the lesion in the dorsal or the ventral spinal cord, as you can see here, that the rats has a difficulty to walk. And based on the dorsal or the ventral spinal cord lesion, you can find differences in the postnatal recovery days. In the later walk, as you can see here, that the measurement is the missing or the slip of the hind paw on the rungs. You can also create these trajectories where you can see a clear visual differences that how the correct functional paw placement is looks like in this stick view. And based on the lesion you have created, either the dorsal or the motor cortex, you can see some subtle changes using either the lesion or the baseline animal. Sorry. In the weighting module, sorry. So this is how when mouse walk, and this is how it's called weighting. So they are walking torso high. So they have to show a lot of muscle strength. But when the ventral spinal cord injury animals, because they don't show this movement, so they cannot walk, right? And you can have, again, these subtle changes being on where you have lesion. But these animals which cannot walk on the wading modules, if you put it in the swimming, they can show a nice stroke movement based on the spinal cord injuries, right? And through these trajectories, you can also see clear differences that this is how the fine stroke movements look like in the baseline animal, where no lesion was occur. But with the dorsal or the ventral lesion, as you can see here, that in the ventral lesion animal, it's completely disturbed. So they don't show any fine stroke movements. In the swimming, as you can see, the tail has a very significant importance. So with after 28 of the post-day recovery, as you can see that these they develop again this sinusoidal wave format. So this also you will see with the Carmelo's presentation, that how this can be very important parameter to detect some motor disabilities, right? So this was being said was for the rats, but here this is also for mice. The motor can be also used, the swimming module can be also used in mice. So this is also a nice picture or nice video. I got it from Carmelo. So this is also very useful. And moderator is for the one device which can be used for rat and mice both. So moderator can be, so hope I explained and convinced you that it can be useful for the phenotyping for all different kind of mouse or the rat models. So it's an injury or neurological disease models or any neuromuscular disease models or simple aging. It can be used for different drug efficacies and for the longitudinal studies. With all of these modalities, it can enhance a lot of translational value, because our software SimiMotion is also used for humans, which means you can have a direct clinically relevant endpoints compared to their humans. So, which can be then also by using appropriate mouse model for the different neurological disease, you can directly compare and both phenotypes between the two different experimental models. You can have also very early detection before the disease progress. So you can identify a specific earlier, any therapeutic substance which you want to test on. So it enhances the confidence overall in therapeutic cases, especially in this preclinical studies design. And our artificial intelligence AI markerless tracking will help you a lot, and it's reduced a lot of working labor. So this will definitely give you a bit more easy time. Our software also is very intuitive. As you can see here, you basically just identify your anatomical points, create your video, and then you can have multiple algorithms which can be based on this anatomical tracking. You can put all this algorithm in one envelope, and this can be your template for. So each of your experiment is a template. So you can just change the video, apply or import all the specific points which you are measurements. You can import it from the previous template and just track again the video. As you can see here, that this the cursor which goes from frame by frame, and you can animate each and every window. So for the stick view or any anatomical coordinates in one video, you can convert that and you can use it for different presentation purposes or for publications. This is the stick view I meant. So you can create also animation on that, and you can see clear visual differentiation. For example, here the DMX, 5-month-old DMX mice, which shows the progressive muscular dystrophy and how there's a clear difference between the wild type versus the MDX mice. You can also see just one stride cycle that how this is differ between the wild type and MTX mice. This is with Huntington disease mouse model, RX2, 10 weeks old, with show a rapid progressive phenotype, wild type versus the diseased animal. So it's a very clear visual differentiation. You can see also pharmacologically treated, cupurazone treated animal for MS, shows a very clear difference, or simple pharmacological agent like diazepam 1 milligram per kilogram, which shows a clear difference that how the diazepam treated animal do have a different motion cycle compared to wild types. And you can see here just the focus on the tail or their snout, that how is differ the length or the height with the wild type. Being said, I'm repeating again that motor radar coming with the 4 different modalities, so horizontal ladder, walking, wading, and swimming. And that you can combine with a TTL box with the third-party measurements like EEG, EMG, or optogenetics. And I want to conclude here that the moderator is the first and only device which allow you to access of 4 different locomotion modalities in one video, allowing a rodent fine motor kinematic gait analysis with more than 100 parameters, so which increase the translational value. Fine motor kinematic gait analysis with moderator reveals subtle phenotype patterns not detected by any other gross motor assays like rotarod, open field, or any other classical conventional test available in the market. It is more sensitive and detecting early gait changes than the current motor test. It's an innovative device, highly standardized, delivering a solid and trustful data, as you can see in multiple publications, and our customers are worldwide in academia or pharmacological industries or any core or the several core facilities. Being said, I would like to thank you, and I'm open for your questions.
Thank you very much, Dilip. That was an excellent presentation. Do want to remind everyone, if you do have a question for Dilip, please enter it into the chat function as we'll be running through the questions as they come in. Let's see here. First question for you, Dilip. What is the minimum sample size required for using the moderator system for preclinical studies?
It depends on the study design, but roughly I would say 5 to 10 animals per group.
Okay. Thank you. Let's see here. Next question that's come in. How long does it take to set up and calibrate the moderator system?
So usually moderator system, so because there is stationary camera mounted on the tripod, is pre-calibrated or factory calibrated. So moderator camera do have an objective, fisheye objective lens. So you have a little distortion on the sides, which is factory calibrated. And there is a procedure which is defined in the manual or provided also on the, it's a soft copy. So if you don't change the height of the moderator camera, you don't have to recalibrate that. So, but if you do, max 10 minutes it takes for the distortion calibration as well as the runway calibration. So roughly 10 minutes.
Okay. Next question, Dilip, has come in. What is the accuracy of the moderator system for measuring gait parameters in rodents?
It also depends on the study design and the kind of anatomical markers you are defining for your study, for your aim and objective. So you can either put all the anatomical markers, and the accuracy with the calibrations you can detect, for example, distance roughly 5 millimeter through the camera, and the angle and everything is quite accurate. And only I would say because your feet is in the motion, so as I said, it's a partially automation. So some of the things which you have to correct manually on each frame if the marker is missed. So I would say 70% you have to is automatically detected, but you have to do 30% work manually on each frame, a couple of the frames.
Okay. Another question that has come in, it says the different functions, parameters that you showed with different programming, they want to know, can we run all of them with only one video?
Yes, this is also depends on if you have marked all the anatomical points, which is possible in that video, then of course you can do that. If you are marking only the hind paw, then automatically you can detect that, but there is also possibility to click all the anatomical points. If even they are not marked, you can mark that manually on each frame without any marker. And that can be then analyzed also. So basically in each video, you can do that.
Okay. I think we have time for one more question. And so the question is, can the moderator system be integrated with other physiological measurement systems?
Yes, as I already said, that there's an optional TTL box, which provides an output or take to the third-party devices to start any physiological recording. So EMG, EEGs, or for example, if you are using force plate, it is not in moderator, but if you can inbuild a force plate below the runway, of course, you can measure the force plate as well.
Okay. Thank you very much, Dilip. If anyone does have further questions, please include them in the chat. What we'll do is we'll include all the questions and provide answers for everyone in the follow-up email. So now I'd like to introduce our second speaker for today, Dr. Carmelo Bellardita, who is an associate professor at the University of Copenhagen in Denmark. Carmelo got his PhD in neurophysiology from the University of Milan, and currently his research interest is understanding the neural mechanisms by which neurons and neural networks operate to generate movements, and how these mechanisms change after traumatic events with a focus on the spinal cord. So today, Carmelo will be talking about behavioral profiling for dissecting brain functions in health and disease. So Carmelo, I'll let you go ahead and take the floor.
So thank you, Gary, for introducing me. I will share the screen so you can see presentation soon. And as Gary said, I'm Carmelo. I'm an associate professor at the University of Copenhagen. And today my talk is gonna focus on behavioral profiling. And literally, I'm gonna actually use all or most of the parameters that Dilip introduced already to you in the moderator. And I show you how we can use this for dissecting brain function in health and disease. But to understand how we do this, I give you just a brief overview of what I do, and then we will go more in detail in the core part of the talk. So as you know, and as Gary said, I focus on movements, and movements comes in many forms in the human and animal existence, and there is literally no human or animal existence without movements. If you think about effortless action as walking or even grabbing a cup of coffee, these are delicate activities that nevertheless represent some of the most sophisticated accomplishment of the mammalian motor system. Now, the mammalian motor system is widespread in the brain and the spinal cord. So literally, we have a network that spans from the brain to the spinal cord to generate movements. So most of our movements are generated by network located in the spinal cord here, and this executes the movement. So spinal networks are active in any moment of the movement, but they are highly modulated by supraspinal structure since movements by definition are goal directed, so implies that we engage by wheel movements, and they are by definition defined temporally. So we have a beginning of the movement and an end of the movement. So you can think that a supraspinal structure like the motor cortex, they are more engaging voluntary control of the movements, while deep brain structure in the midbrain and brainstem, they are involving the initiation and stopping of the movement. Now, since the movement is the only physical output of the brain, you can understand that spinal cord injury, which is clearly a traumatic events that happen to the spinal cord, disconnect the supraspinal control from the control of the spinal cord, and this brings to motor paralysis, so to lose the possibility to engage voluntary movements. So all my work, then you can understand that it's crucial for me to identify and describe movements for understanding how the brain generates movements or motor movements, and how then we can provide a therapeutic solution to recover locomotor function. So today we are going to focus on this type of features that are going to be here, and they are going to be all implicated in describing movements in mice. And we're going to go through them using my talk. And my talk will be divided in three parts, where we show three projects that heavily use behavioral profiling. And at the end, we will again have the list of these features that I just showed you, and understand how we use them to understand how the brain control and generate movements. So we will begin defining as with a gait analysis that we ran some years ago in the lab, where we use the motorator to define gaits in mice. So as you may know, gaits are literally way in which mice and most of the quadrupedal mammals can move, and they coordinate limb movement. And there are different gaits, so animal can use different way to coordinate limbs, and usually the difference between them allow the animal to increase the speed of the movement. So depending by the context and the needs of the animal, they can change gaits to increase the speed of their movement. Especially in mice, today we're going to see four main gaits: walking, trot, gallop, and bound, that characterize, and these are the main, most exhibited gaits in mice, and they allow the mice to move from slow walking, for example, when mice are exploring new environments, to high-speed movements, like gallop, that is used by mice to escape from predators. So this is, for example, a mouse that running in the motorator in the corridor with one single view, in this case, just to simplify. And you can see that depending by the speed, it can have a slow walking, trotting, and then increasing the speed, galloping and bounding. So what we have done here was clearly to, since speed is the measure parameter that can tell us which gait is using the mouse, we describe the speed of the mice during the different gaits, and you can see from walk to trot to gallop to bound how the speed increase. And speed is mainly clearly controlled by two components, which is the step frequency and the stride length. And both step frequency and stride length, they are clearly increasing as long as we move between gaits, and they can be clearly seen as a relation between step frequency and speed, or stride length and speed, and see how they participate in determining the final speed of the animal. However, speed do not define gaits. As I told you before, gaits are defined by limb coordination. So we need to understand more how limbs move to understand which gait the mouse is using. And to do that, I will briefly explain you what we use to characterize interlimb coordination. Now you can see that this is a step of a human walking, and this is the next step. And when the animal or the human clearly are touching the ground, this is called stance phase, and when it's lifting, it's called swing phase. Now, in this case, we have a step cycle, which is 2 consecutive stance phases, and give us the possibility to, for example, calculate the stride length or the stride frequency. Now, in this case, we are going to compare one foot to the other foot. And in this way, we can see literally when two foots are simultaneously on the ground, in this case, they are defined in phase. And you can think when, for example, we jump or we skip with both feet, that they are synchronized. On the other side, for example, when we are walking, we are out of phase with the limbs and we are literally alternating them with one stance and a swing, and then we invert the swing and the stance for the feet. So there is a mathematical way to clearly quantify the phase of two limb moving, and this is usually used with the circular statistics and the circular plots. In this case, if you look at the step cycle, in this case here, of 1 feet, that is going to be our reference feet, is going to be normalized from 0, the beginning, to end to the next, which is 1 to the next phase. We can plot this as a circular plot where 0 is the beginning, and then we go around to see the step cycle in 1, and then we're going to plot the other foot in relation to the phase. So when we are going to have 0.5, so it's in the middle, we are clearly steps that are out of phase, or rather alternating. When they are going to be on the other side in phase, they're going to be at 0. So this is going to be an easy visual graphical representation to understand whether two limbs are synchronized or alternating. And now we can go and quantify better, define the gaits in mice. So in this case, we have walking, which is usually used in mice for exploring new environments. So it's extremely slow. And usually you can see here that usually he puts the feet on the floor in a specific order, with one forelimb, then the contralateral hind limb, then the ipsilateral forelimb, and then the contralateral hind limb, in continuously in this step cycle. And we can quantify the moment that the stance phase in darker here and the white and the swing phase last during the step cycle, the normalized step cycle for each of the limbs. And then we can also look at the, clearly, as I told you before, at the coordination between the limbs. In this case, we have our reference limb, which is the left hind limb in black. And then we look at all the other limbs, and you can see with that the corresponding hind limb, it's alternating. The forelimbs are alternating between them, and they are slightly shifted in time compared to the hind limbs. Now we can combine this information, the phase, together with the stance phase, and we obtain this graph where we have a step cycle, and then we can see simultaneously that the stance phase of 3 feet is on the ground. So we have 3 feet on the ground, which is exactly the definition of walking. So this helps us to identify walking as a gait. And then we have done the same for nearly all the gaits. And in this case, we have trotting, which is characterized by a diagonal coupling between the forelimb and the contralateral hind limb, and then again, the forelimb and the contralateral hind limb. And this is possible because in this case, we have a short stance phase that allows more or less 50% of the step cycle and allows the other limbs to get, and then the coordination between the limbs is clearly the two hind limbs are alternating, the two forelimbs in blue and green are alternating, but you can see that one forelimb and the contralateral hind limb are simultaneously moving, and the other two is the same. So when we combine stance phase and coordination between limbs, you can obtain this step cycle where you see the two limbs are simultaneously in stance, and then they leave this for the other two limbs to be in stance. And this is exactly how trot is defined. And then we move to higher speed gaits, where in this case, mice show a synchronization of at least the hind limb movement. So you can see here that in this case, hind limb movements are more synchronized and they start having a simultaneous swing phase. And you can see better here in the video, where the animal have both feet on the hair and then sometimes touch the floor together with the hind limb. And also in this case, it's possible because the stance phase became even shorter and we have a long swing phase. In this case, we have the gallop where the two hind limbs are perfectly synchronized and the two forelimbs are still alternating. Now, if we combine again the limb coordination together with the stance phase, you can appreciate here how you can see that each of the limb goes on the floor in a sequential way with the two hind limb simultaneously on stance phase and then simultaneously on swing phase for at least a part of the step cycle. This gait, it's clearly introduced a new part that is called the ideal phase, where for a moment all the limbs of the mouse are on the air and the mouse is literally jumping. And then when the forelimb synchronizes well, we move to bound, and you can appreciate here where the forelimb are touching simultaneously the ground, and the high limb too, so they are perfectly synchronized to each other. And even though the stance now doesn't change a lot, is the coordination between the limbs that changes, having the high limb coordinated and the forelimb perfectly synchronized between them, and introducing two ideal phase, one during the high limb, the forelimb, and one during the high limb stance. So in this way, we have looked at the interlimb coordination. Now, one important part that we clearly are interested in the lab is how the brain may generate these gaits. And a first idea of how this may happen, so how the neuronal control of movement can generate and maintain gaits, is clearly given by muscle activity. And to get an idea of the muscle activity, we use the intralimb coordination. So you can see that this is, for example, the high limb of a mouse. We have a segment, the thigh, the shank, and the foot. And currently this part can be analyzed with the kinematic analysis, where we have the stick diagram decomposition with the 3 segments. And clearly during the frames, so during the video, for example, during a step cycle, the movements of the limbs have a certain, in this case, for example, forward movement during the lift. And you can see that all the 3 segments move in a flexion way, so we can in some way guess what is the muscle activity at this level, because specific muscle moves specific joints, and we can know whether flex or extension activity is going on. So we can use body segments as a proxy for muscle activity. And in this case, we have done clearly, we have examined specific components of the movement. In this case, the first thing that we see is the angular excursion, which gives us an idea of the spatial components of the step cycle. And we are going to measure the maximum angular excursion of this animal during the gaits. And as I told you before, as long as the gait, the mouse change gait from walk to trot to gallop is increasing speed, and you can see by eye here during the kinematic analysis of the honey, that clearly the stride length by eye gets bigger. And how this may happen is clearly generated by the different joints that are used, or the different body segment, in which the maximal angular excursion increases as long as increasing speed. So now to have a brief quantification of this, you can see that for each joint, mice change, have specific patterns, depending on which gaits we are looking at, but clearly, as long as the mouse increases in speed, each joint increasing amplitude. You can see, for example, when we talk about the thigh, how it's increasing during the 4 gaits, and the same for the foot, and the same for the shank. So what is true is that each joint, when the mouse reaches the highest speed, is at the highest angular excursion. On the other side, we can clearly look at the spatial information component, but we would like also to look at the temporal component, since it's crucial to understand not only what is the maximum angular excursion, but how this can change in time. And in this case, we are going to look at the maximal angular excursion and how long it takes, for example, to increase the angular excursion of the thigh during the stance phase, during the swing phase, and then when it goes back to the stance phase. So we can look this during the step cycle, and we look at this for all the mice during the different gaits in walk, trot, gallop, and bound. And you can see that here, each of them has its own specific pattern. So each gait has a specific temporal pattern for the joint movements. And as long as the mouse increases speed, each body segment became more synchronized with the others. So you can see here in walk where literally each joint is actually having a delay compared to the previous one, but you can see here the 3 joints that actually almost are synchronized when the mouse is having high speed movements. So in this case, here is the first part of the talk. Now we start with this first part. We look at specific parameters like stride length, stride frequency, and speed. Then we look at the interlimb coordination to look at the homologous diagonal epilateral coupling between the limbs, and this was useful for us to define gaits. And then we move to intralimb coordination to examine the spatial and temporal components of joint movement and begin to have some idea or some indication of how muscle activity might control and generate movements. Now, in the next part of the talk, we will relate movements to specific brain activity. In this case, will be specific spinal activity. And I will show you how some specific spinal circuitries mediate and generate specific movement. So as I showed you before, I show you that mice move with different gaits, but clearly walking is not the only locomotor strategy that mice use. they change strategy depending by the context in which they move. So in the motorator, as you can see here, you can have different tasks, and depending which task the mouse can change is locomotor strategy. In this case, for example, is skilled walking on the ladder, and the mouse has to be much more precise in when it touches the sticks there. And then this is for example, walking on a rope, where the animal has to balance the body in a much more unstable environment. And then we have swimming, where the mouse has to literally move in water, and then he has to use the body in a different way to propel himself forward. So what we have done in the lab was to literally examine more closer the kinematics of the animals from a side view and from a bottom view. In this case, we focus on 5 different tasks. So walk, trot, and gallop that we just went in detail to see a few minutes ago. And then we swimming and rope. And this is the ventral view of the mouse during the same task. And then we use the stick diagram decomposition to examine more deeper some specific body segments, like the hind limb, right? In blue, the forelimb, and then the trunk in gray with the tail. And you can appreciate how the mouse how he changed the coordination of each body part to change the locomotor strategy. But nevertheless, during the stroke cycle or step cycle, which is our reference point in this stick diagram, still of the body segment are well coordinated between them. Now, if we just focus, for example, on the tail, you can see and you can appreciate how the tail is differentially modulated during each task, implying a strong neuromuscular control of the tail from the brain and the muscle. So we are going to focus on this particular body segment, the tail, to examine some brain function, in this case specifically how spinal circuitry may mediate and may generate the control of the tail during different tasks. For who is not familiar with the tail of a mouse, the tail of a mouse is literally done by multiple vertebral bodies that are repeated through the tail, and each vertebral body is linked to the next vertebral body using 6 muscles, 2 on the dorsal side, 2 on the medial, and 2 are on the ventral part of the tail. And these 6 are repeated along the tail. Each muscle, then it's composed by intermingled muscle fiber, with fiber of type 1, type 2A, type 2B, and type 2X in different proportion that you can see here. And literally, with this anatomical architecture and with the fiber composition of each muscle, literally the tail can be controlled to move in any direction and to support explosive movement, like can be required when the mouse is escaping, or to support prolonged activity, like when the tail is lifted up when the mouse is walking. So we decided to enter and use this as a model to study how spinal circuitries control movement in the tail. And to understand this, we decided to ask first how the spinal cord can control tens of muscles whose motor pools are located and segregated in the spinal cord, and they have to have distinct function, for example, agonistic and antagonistic muscle to move joints. So we begin to answer this question with the specific population of neurons, of interneurons in the spinal cord that are characterized by the promoter CHOX10. And we use the mouse intersectional genetics to identify our interneurons in one single segment of the spinal cord, and we inject a viral vector that expresses tomato in the mouse, in the neurons of interest, and GFP in the terminals when we inject a viral vector upon Cre recombination. So we can look at actually at the projection pattern of the cell, and you can see here that most of the synaptic projection of this population is in the site where the soma is located, so where the site of injection was, but they have a strong projection in more caudal segments of the cord, generating a strong feed-forward excitatory system along the rostrocaudal part of the cord. Clearly, we also look at specific connection of these specific interneurons, especially their direct connection to motor neurons, so that they can control muscle contraction directly. And then we look at the specific connection to neurons of the same type. And these neurons are glutamatergic neurons, so we can have a V2A connection. And then we look also at the connection of this neuron with inhibitory neurons, labeled by GlyC2 in our mice. But we wonder, what is the functional meaning of this connection? So to answer this question, we use again mouse intersectional genetics and electrophysiology in vivo together with optical stimulation to probe the connection of this specific connection. So what we have done was to use again a mouse intersectional genetic using our Chuckstein-Crick mice, and coupled with a reporter mouse that express channel rhodopsin under the Chuckstein-Crick promoter. So we can label all our interneurons and they can express the opsins, the channel in the cells, in a way that when we stimulate, when we shine a blue light on the top of the cells will depolarize and they will activate the motor neurons that in turn will activate the muscles. And we can record the muscle and see the monosynaptic activation based, clearly monosynaptic based on the latency. So we can study how these cells control muscle activity. And this is a typical example of the study we conduct where we shine the light on the spinal cord along the rostrocaudal gradient of the spinal cord, and we simultaneously record one cell here, and we can record clearly multiple muscles. And in this case, for example, you can see that when this cell is active, this muscle is also active. But then there are other two that are active when actually the cell is silent. So this is indicative that these cells may actually be involved in driving directly this muscle, but not these other two. And based on this type of analysis, what we have done for clearly, first we look at the projection between V2A neurons and motor neurons, and we look at how these cells may control muscle activity. And what we have done first was to stimulate the spinal cord in the most rostral position, and then we record 3 muscles at the base, at the middle, at the tip of the tail, and then 1 contralateral muscle. And you can see that when we shine the light in the most rostral part of the spinal cord, we have a muscle contraction in the most rostral part of the tail at the base. But then clearly, when we move more caudally, our optical stimulation, we contract the muscle that is in the middle of the tail. Here you can see. And then when we move even more caudal in the spinal cord, we contract an even more caudal muscle in the tip of the tail, indicating a rostrocaudal gradient in the innervation of the cell of the V2A cells of the motor pulse. And even more striking here was that when we stimulate a lateral side of the spinal cord, we get the contraction of a ventral muscle at the base. And then when we stimulate more laterally, more medially in the spinal cord, we get a contraction in the medial muscle of the tail. And then when we move again more medially close to the central canal in the spinal cord, we induce the contraction of only the dorsal muscle. So this experiment showed that there is almost a topographic musculotopic representation between the location of our cells and the muscle contraction, with a fine globular innervation of motor pulse by the Chuckstein neurons. However, in this case, we can clearly activate single muscle by a specific stimulation of single neurons. But how we can generate the activation of multiple muscles that are required, for example, during swimming, starting from one single population. So we thought that to activate specific multiple muscles, we need to use probably multiple pathways that are controlled by the interneurons, but they are polysynaptic. So in order to do that, we checked first the first connection, which was V2A, so excitatory neuron to excitatory neuron that can help to diverge the motor command and recruit multiple muscles. So what we have done for this experiment was to record the neurons, the V2A here, and the muscles, and then shine the line on the V2A. And you can see that we can activate the cell and simultaneously activate the muscle, the M3 in this case, and the latency between the two tell us that this neuron is activating with the short latency and monosynaptically the other muscles. Now, if we move the more rostrally, the optical stimulation, what's happening is that in this case, the activation of our neuron is delayed. You can see the latency compared to before, but the latency between the cell activation and the muscle activation is similar. So this, we have to imply that this cell is activated by another population more rostrally, at least with a monosynaptic connection and even more maybe. In this way, what we conclude with this experiment is that we found how a single population of V2A can control multiple muscle by recruiting other V2A more caudally in the spinal cord, and then they can control more muscles. Now, in this case, these are all excitatory circuitries, so they're going to all activate muscles, but when we move, we need to quickly activate some muscle, but then inhibit others. So we need to have some kind of functional inhibition on the antagonistic muscle to allow a joint to work and then move clearly forward or backward. So to do that, we search for functional inhibitory cells that may be recruited by the V2A in the spinal cord. And to do that, what we have done was similar to before. We stimulate the cell and we record the cell in this case, but you can see that in this case, the cell do not respond to the light. And so it's indicate that it's not a V2A cell. On the other side, when you move the light more rostral, what happens is that the cells start responding with firing action potential, indicating that the stimulation can recruit these cells. And curiously, when the cells start firing, here, for example, you can see the muscle, the corresponding muscle was silenced. And the silencing of the muscle was proportional to the firing frequency of the cell. So the higher the firing frequency, the more the muscle was silenced. So in this case, this indicates that our V2A cells that were optically stimulated here more rostrally can recruit inhibitory network more caudal to them, and then this inhibitory network can silence functionally the muscles. So now we have a system that can clearly recruit multiple muscle with functional distinction. However, how this can recruit multiple muscle in a sequential order, because clearly when we walk, our muscle are gonna be recruited sequentially to generate a step. And on the other side, with a different amplitude. So each muscle has its own amplitude and it's gonna be clearly related to the function of that muscle in the movement. So we thought that this timing and amplitude may be regulated by some sensory feedback that each muscle has, and is specifically called proprioceptive feedback. And to test whether our interneuron can receive this input and then integrate in the stimulation of the muscle fiber, what we have done first was to look at the anatomical evidences for this connection. So what we have done was to inject cholineratoxin beta subunit in the muscles that will be retrogradally transported in our sensory neurons. And what will happen is that we will see synaptic boutons of the sensory neurons on the top of our cell of interest, in this case, the CHOC stem. And you can see here that actually we have sensory boutons on our cells. So this indicates where we have sensory input directly on the soma of the cell or in their dendritic axons. And then we also clearly check if this sensory input were functional, and then what we have done was to stimulate the sensory afferent with low threshold, and then record the cell and the motor neurons, and we can see that the stimulation of the sensory axon evokes an action potential in the cell whose latency is compatible with the monosynaptic activation. So now we know that they receive sensory input. We want to test whether the integration of sensory input from our interneuron may generate the muscle sequence activation. So to do that, we have recorded 3 muscles. And first of all, we stimulate we stimulate the sensory afferent of one of the muscles, in this case M2, and you can see that we get a response here in 2 muscles, M2 and M1 that was just directly above, but not M3. Then we did the optical stimulation of the CHOC stem neuron. So in this case, we are going to stimulate our interneuron optically, and you can see that they are going to drive the activity of M2 like before, but also M3 that before was not activated. So indicating that these muscles and these muscles do not have sensory input, and this muscle doesn't have the integration of the CHOC stem neurons. But when we combine them, we can see that we get a sequence of muscle activation with different timings and with specific amplitude for each muscles. So now we have a sequence of muscle activation with different amplitude. So we know that this system may be useful for generating sequence of muscle activation. On the other side, the sequence of muscle activation, we are talking about in the order of 50 milliseconds, is too short to be relevant to at the behavioral level. So we want to test whether this may be sufficient at behavioral level to control a movement. And to do that, we decide to investigate the action of this circuit in a specific behavioral task. In this case, swimming. Why swimming? Because, and you can see here the mouse in the motorator, with the side view on the top and the dorsal view on the bottom, how the mouse swim. You can see that they are amazing swimmers with the tail that move left and right, and you can see there for the dorsal. And then we have strokes from the hind limb, and the forelimb are mainly inactive, or they do these directional strokes just to correct slightly the body posture. Then what we have done was to silence our interneurons using, again, a viral strategy to deliver tetanus toxin. So once this will be picked up by crine neurons, in this case our spinal interneurons, they want to have the possibility to release glutamate, so they will be functionally silent, still alive in the spinal cord, but silent. And then see how this silencing affects the tail movement and the entire swimming. And what happened was striking, because clearly the tail did not move at all, almost paralyzed. And the fact that the mouse can use the tail to propel himself corrupt the entire locomotor strategy, and then you has to, the stroke of the hind limb, and in particular the forelimb, has to compensate for the paralysis of the tail. And clearly we quantify this with some parameter, especially the speed, since the tail clearly is important for the animal to speed up and propel himself in the water. And then we look at the temporal part, where the tail is moved together with the rest of the body in a sequential activation, where the tail oscillates. And you can see that during control in our healthy mice, that the tail oscillates together with the rest of the body. Here, and in blue, you can see the tail, all the tail segment did not oscillate anymore, indicating paralysis. And since the tail do not oscillate anymore, even the amplitude was reduced greatly compared to the control. So for this study, we use and we look at the body coordination, and in this case, clearly the spatial and temporal components that may be important for other body segments, including the head, the trunk, and the tail. And we look how we can perturbate or record the neuronal activity and associate the neuronal activity to specific behavioral outcome. Now, in the last part of my talk, I will focus on how we can use clearly deep behavioral profiling for understanding how we can restore movement after spinal cord injury in our mice model of spinal cord injury. So as you know, and as I showed you before, the motor control system is widespread in the brain and the spinal cord, and clearly, as I told you before, there are some deep brain nuclei that are crucial for initiating movement. So in the lab, about several years ago, we have stimulating a specific brain nuclei called the cuneiformis using optogenetic. And when you stimulate this area, the mice can start walking, but if you increase the stimulation frequency to 10 Hertz, the mice start walking. And then if you increase to 20 Hertz, mice start trotting. And then when you go to 50 Hertz, mice start galloping. So the stimulation of this area, it's able to evoke all gaits in mice from the low speed to the high speed. So we thought that this may be extremely useful after spinal cord injury, when the animal is not able to walk anymore, but still there is usually a spinal cord injury is incomplete, and we can maybe use the spare descending fibers to drive locomotion. And this is what we have done here. So this is a mouse that has a thoracic injury, and you can see here that the limbs, the hind limbs are paralyzed, and the animal currently is able to use the forelimb to walk. And this specific lesion has destroyed the dorsal part of the spinal cord and the gray matter, but leave intact the ventral funiculus, where most of the descending fibers that are used to activate the spinal circuit to walk are intact. And you can see here in this part, especially, that the ventral part has the reticulospinal neurons that drive, that are important for the pre-measure rule in triggering and maintaining locomotion, are intact, while from the rest of the cord we have a complete absence of fibers. So, and then what we have done was clearly to, again, use the cuneiformis nucleus, inject a viral vector to deliver channel rhodopsin to the cells that are in the cuneiformis nucleus, and then implant a fiber to deliver the light, and then see whether our mouse, that is the same mouse that you saw before, the paralyzed mouse, can walk or can instantaneously perform a better motor performance. So you can see here in the video, when the light is blinking, then the mouse increases speed. And then if we really focus on our main point, so the hind limb, and we run a kinematic analysis in the step cycle, specifically looking at the foot trajectory, you can see that the foot trajectory change between the light off, in which we have a step that is extremely short and low, to when the light is on, in which the stride length is increased and also the stride height is increased, showing how the stimulation of this specific nuclei may acutely improve the motor performance of the animal after injury. And with this, I show you how we can couple clearly optical stimulation to behavioral profiling as a cornerstone for preclinical application and see how we can actually have a translational potential in biological discovery that may be used for as a therapeutic solution for motor impairment, like in this case for spinal cord injury. And with this one, I would like to conclude and show this work has been mainly done at the Copenhagen University of the Karolinska Institute in the lab of Ulrik Kien, together with my colleagues. And clearly, thanks for your attention. I may like to leave the last slide with our features that we show today during this, in case of some question may be relevant for this. Thanks very much.
Thank you, Carmelo. That was an excellent talk. Again, thank you for the information that you have provided for everyone. We do have some questions for you. The first question is, you looked at the movements of the limbs in the first part of your talk and the body movements in the second part of your talk. However, when mice walk, all body parts should be in some way coordinated. Isn't that correct?
It is, yeah, clearly I split them in two parts because clearly it's easier to illustrate for people to describe, but if you clearly think that gaits, for example, imply interlimb coordination, but then also the rest of the body is clearly well coordinated with the limbs, you can clearly run them all together and see how limbs may be used as a reference point to see how the rest of the body moves during the step cycle, because the step cycle, it's easy to measure clearly and evident when we look at it, but it's definitely correct and may be useful for many investigation at the behavioral level.
Okay, thank you. Next question, which type of statistical tests do you use when comparing data from different groups of animals?
So, I mean, at the beginning, for example, when we use stride length, stride frequency, these are linear measurements, and we use simple statistical analysis, like you can use a t-test if you have two groups, or one way or another if you have more than two groups. But for example, when I look at the limb phases, for example, these are clearly, or angular excursions, these are radiant or degree data, so you need to use kind of a circular statistics. So instead of using the t-test, for example, you can use the Watson U2 test to measure statistical difference between these groups. So it's only to pay attention in case you are using circular statistics for this type of data.
Okay, next question that has come in. Do you have any suggestions on how to optimize the video for tracking the animals in different conditions? For example, when the mouse is swimming.
Oh yeah, so this is a kind of an important part, and I think I saw some question also before. Clearly, tracking is one of the major part of this work, and it's important to optimize tracking, especially, I don't know, for swimming is difficult because clearly you have water, and the water diffract lights differently from air. So when the mouse is moving, there will be some white spot on the video where definitely can be difficult to track any body segment there. So you may need to specifically control the light during this task, swimming, or and optimize this, for example, using specific angles where the light can avoid to diffract and catch the camera.
Okay, I think we have one more question for you, Carmelo. Did you train the animals before testing them?
In the case of the gaits, for example, I did not, because while maybe high-speed gaits can be evoked by, I don't know, escaping, unfortunately for walking, which is a really slow gait, was extremely difficult to obtain this if the mouse get used, and then this is why you train the animals so to get used to the test. But if they get used to the test, they do not exhibit slow walking. So in that case, I did not. But for example, in the case of spinal cord injury, when the animal need to go and be tested several times along the observational period, then I train the animal 1 week for again more or less a week, so they can get used to the test and they do not change, or the changes that they show are clearly dependent more by the lesion or by some neurorehabilitation. So in this way, I can discriminate better. Okay.
Again, Carmelo, thank you very much for your talk. Really appreciate the information that you were able to share with us and your experiences with the Motorator.
It's been a pleasure. So no problem at all.
All right, everyone. Just a few things as we conclude our webinar for today. I do want to ask all of our participants, when you have a chance, please fill out the questionnaire that we will be sending to you to help make our future TSE webinars even better. The link will be sent out directly after the session from today. You will also be receiving a recording of the webinar, as well as all the questions and answers that were asked today after each presentation. That will be sent to you a few days after today once we've collected everything. If you do have any additional questions or would like any additional information on the moderator or any other TSE products, please contact us directly at info@tse-systems.com. And of course, for those of you that aren't doing so currently, we invite you to follow us on our social media, on our LinkedIn and Twitter accounts, in order to stay in touch with the latest news and information from TSE Systems. So with that being said, again, I would like to thank Dilip and Carmelo for their presentations today. And of course, I'd like to thank everyone in the audience for participating in this webinar event. We look forward to seeing you soon in our next webinar event that will be coming up shortly. So take care, everyone. Enjoy the rest of your day. And again, thank you for being a part of this event.